So let's go ahead and we're going to continue our study on the nucleus function. So let's go ahead and study that so we can understand that our bodies are fearfully and wonderfully made. There's two main functions of the cell nucleus is to control your gene expression. In other words, everything in that nucleus, it tells you exact everything, the blueprint about you is in that nucleus. It also mediates the replication of DNA. We know DNA means deoxyribonucleic acid. Deoxyribonucleic acid. It is short. The short way of saying that is DNA. So it talks about, it mediates the replication of DNA. So your DNA has to be replicated, has to be copied. And God has a way to copy your DNA to make sure every single cell has your DNA. Every cell has your DNA during the cell cycle. So what is gene expression? What does it mean by gene expression? First, to get a gene expression, in other words, to read what's on that DNA so it can express and do what it's supposed to do. The first involves transcription, very important, in which DNA is used as a template to produce RNA. That's a close cousin of DNA, and RNA is ribonucleic acid. And basically what it does, it copies like a Xerox machine. Transcription, it copies like a Xerox machine DNA. And the result, after it copies, is RNA. And RNA is the product, is a produce. That's what it's produced. It produces RNA after it goes through transcription. In the case of gene encoding proteins that RNA produced from this process is called a messenger RNA. So once the process is complete, once it's done, that messenger RNA goes outside of the nucleus and goes and attaches itself to a ribosome, which is located outside the nucleus, and we'll see that in a little bit. There's 3 steps to protein production. Protein is the raw material that makes up parts. But before you have the raw material, you have even rawer material that makes up protein. You got material that makes up protein, and protein makes up parts, or makes up different things that we need. Like you may have hemoglobin. Hemoglobin is made through this whole process. Hemoglobin is needed on your red blood cell for it actually to receive oxygen. So it goes through this 3-step process. The first process is replication. In other words, all your DNA needs to be copied. It's duplicated through a whole process of meiosis, mitosis, cell division. So you have an exact copy of your skin cell replicated all over the body, all over your skin. And you have replication of muscles and stuff. But first of all, you have replication of the nucleus to make exact copies of its DNA. So you have replication, but we'll go through that another time. But then you have what's called transcription. And transcription, as we just mentioned earlier, is when the process in which a DNA is used as a template to copy all the genetic code on there to make what is known as RNA. And when RNA, ribonucleic acid, is made, it becomes a messenger, goes outside the nucleus, attaches to a ribosome, and actually helps make a part. And we're going to look at that in a little bit. That is known as translation. Translation is when that messenger RNA comes out of the nucleus and attaches itself to the ribosome. But it's used to line up amino acids. Amino acids are the raw material. You got this RNA, amino acids, the raw material of making protein, a protein molecule. And let's look at this whole process. Matter of fact, I'm going to show it to you again, and I'm going to show a video of this whole process. So remember, inside the DNA, because I can't show you that, but inside that nucleus, it's literally copying it like a Xerox machine, becoming an RNA, and that RNA goes outside the body. Goes outside the nucleus and attached to a ribosome. But then once it does that, translation happens, okay? And we'll show you that in a little bit. So you'll see how fearfully and wonderfully you're made. I will praise thee, for I am fearfully and wonderfully made. Marvelous are thy works. that thy soul knoweth right well. Psalms 139:14. So let me turn to that real quick.
What you are about to see is DNA's most extraordinary secret. How a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the 2 strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA.
So that yellow you see is RNA.
The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.
And now we're going to look at translation. This is where they actually they make the proteins. So watch.
When the RNA copy is complete, it snakes out into the outer part of the cell. Then, in a dazzling display of choreography, all the components of a molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. Each transfer molecule carries a 3-letter code that is matched with the RNA in the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecules. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second. And as a result, our muscles, brain, and all the vital organs in our body receive the oxygen they need.
You see, so I'm explaining just a little bit some of the things you saw in the video. Now, the first part you saw was transcription happen. That's not transcription. Well, transcription was literally where it made a little. We'll study it in more detail later, but actually made a copy, an RNA copy of the DNA. A working copy. So when it goes outside, you saw the various enzymes get together and it collapsed. It actually connected to the DNA, and the DNA is like a zipper. It's a double helix, and we'll study that next time. The double helix is like a ladder that's twisted with their It looks like a little ladder going all up, and we'll look at that another time. But it unzips it. That RNA is made through that unzipping process. So when that enzyme goes down that chain, unzipping, just like a Xerox machine, is copying it. It's literally copying what's there and making its own template to go and make some stuff, because it got to know what kind of blood, say for instance, what kind of blood type you are. It's all in that DNA. So like you saw in the example, it made a ribosome. Not a ribosome, it made a hemoglobin. But say for instance, to make parts for your skin, in that DNA is going to make protein for your skin to be made. It's going to unzip it to make sure it got the right color. You see? Or your hair, to make sure it has the right color hair. So it's copying all that information, and then it goes to make the parts. So that's the first part. So the first part is unzipping that DNA to make RNA. And when RNA comes outside the ribosome, it's known as a messenger RNA, an mRNA. And that messenger RNA goes out and it lines up with amino acids. Amino acids is known to actually help make parts. And every amino acid, it makes different parts. It's needed for making different parts of the protein. So that's what we saw in the video. So in that translation part, the translation is when that RNA comes outside. Remember we saw it come out, snake on outside. It attaches to the ribosome. But what's working with it, of course, it goes through this factory, so to speak, making protein. So once this chain comes on down, they have what is known as a, this is the messenger RNA. You see that right there? That's what came outside the DNA, right? Then you have what is known as a transfer RNA, a tRNA, transfer RNA. What it does is carrying amino acids. And it attaches itself, which we'll learn, because there's only, when you look at the DNA, certain letters, we just call them letters right now, can connect to each other. So like in this case, A and U is actually a T from the actual DNA, but they're copied as U, but only they can connect together. An A and a C cannot connect together. An A and a G cannot connect together. A G and a C can only connect together. So what they do is they come down and line up on that chain. So this is the transfer RNA that's carrying the amino acid. And this is these are 20-something different amino acids, and it's carrying an amino acid here, and it carries it on down to make the part, and actually that's what it's doing. It's linking up. It's known as a peptide chain. So it comes down, links up, and it makes that. That's actually the finished product. So once it's done bringing down it brings the tRNA, brings it down, links it up, connects to the messenger RNA, and once it's done, it's outgoing, it's empty. That's all the process. So that's known as peptide synthesis. This whole what's happening right here. This translation. So understand the difference between transcription and translation. All right. So we'll continue to study this next time. So we'll get into, because I didn't get a chance to show you the video, and I just want to repeat what we did today. So you can have a clear understanding. But next time we'll look more in detail of the DNA, the dialloxynucleic acid. We'll look at all that, what it is and how it looks.